kemajuan yang dibutuhkan untuk mewujudkan visi besar di masa mendatang. Layaknya sebuah orkestrasi, Badan Riset dan Inovasi Nasional atau BRIN hadir mengharmonisasikan berbagai kegiatan penelitian untuk pengembangan, pengkajian, dan penerapan, serta mensinergikan riset dan inovasi di Indonesia. Membentuk ekosistem yang terdiri dari 7 kedeputian, 12 organisasi riset, dan 85 pusat riset, yang diantaranya Organisasi Riset Penerbangan dan Antariksa, Organisasi Riset Tenaga Nuklir, Organisasi Riset Energi dan Manufaktur, Organisasi Riset Kebumian dan Maritim, Organisasi Riset Hayati dan Lingkungan, Organisasi Riset Elektronika dan Informatika, Organisasi Riset Ilmu Pengetahuan Sosial dan Humaniora, Organisasi Riset Arkeologi, Bahasa dan Sastra, Organisasi Riset Kesehatan, Organisasi Riset Nanoteknologi dan Material, Organisasi Riset Pertanian dan Pangan, Organisasi Riset Tata Kelola Pemerintahan, Ekonomi, dan Kesejahteraan Masyarakat. BRIN juga mendorong riset dan inovasi menjadi ekosistem di Indonesia yang maju dan berkembang dengan 2 sasaran utama. BRIN menyadari potensi luar biasa putra-putri bangsa dalam bidang ilmu pengetahuan dan teknologi. Untuk itu, berbagai kebijakan riset dan inovasi diarahkan untuk menghadirkan kompetensi SDM iptek terbaik. Menghasilkan berbagai program unggulan sebagai bentuk komitmen BRIN terhadap periset, inovator, serta masyarakat luas dari berbagai disiplin ilmu di seluruh negeri. Peningkatan kualitas ekosistem riset dan inovasi di Indonesia juga tercipta melalui kolaborasi dengan berbagai kementerian, lembaga, industri di dalam negeri, dan pemerintah daerah, di antaranya dengan dibentuknya Badan Riset dan Inovasi Daerah atau BRIDA. Selain itu, kerjasama dengan berbagai lembaga riset di luar negeri juga terus didorong melalui partisipasi aktif sebagai anggota dalam berbagai organisasi dan forum internasional. BRIN menata alur riset sehingga lebih efektif dan efisien dengan mengintegrasikan sumber daya manusia, infrastruktur, dan anggaran dari berbagai lembaga litbang jirap. Berorientasikan prioritas riset nasional, BRIN melakukan evaluasi secara komprehensif untuk setiap percepatan yang tengah berjalan. Upaya tersebut didukung melalui pengelolaan berbagai fasilitas riset dan inovasi yang tersebar di 17 provinsi di Indonesia dan akan terus bertambah mengikuti perkembangan kemajuan iptek. BRIN terus meningkatkan kontribusi riset dan inovasi terhadap pembangunan nasional melalui pembangunan dan revitalisasi 4 kawasan sains dan teknologi atau KST. Sebuah kawasan terintegrasi berupa fasilitas laboratorium riset, laboratorium pengujian, fasilitas industri, baik startup maupun UKM pada skala kecil atau uji coba, ruang kantor tenan, co-working space, ruang pertemuan, ruang pameran, dan fasilitas pendukung lainnya. BRIN hadir untuk mewujudkan cita-cita menuju Indonesia Emas 2045. Melalui konsolidasi sumber daya iptek, menciptakan ekosistem riset, dan fondasi ekonomi berbasis riset. Menjadikan Indonesia sebagai rujukan dunia dalam pengembangan dan pemanfaatan ilmu pengetahuan dan teknologi, kebijakan, serta kemanfaatan bagi umat manusia. Alam adalah sumber inspirasi. Ilmu adalah fondasi. Dan mengamalkannya untuk kemajuan bangsa.
Selamat datang di Pusat Riset Biomedis, Organisasi Riset Kesehatan, Badan Riset dan Inovasi Nasional. Selama 4 tahun, Pusat Riset Biomedis telah berkiprah dalam mengembangkan riset berbasis kesehatan, mendorong inovasi, serta menghadirkan kontribusi ilmiah yang berdampak bagi masyarakat. Inilah perjalanan Pusat Riset Biomedis. Melalui berbagai webinar dan forum ilmiah, Pusat Riset Biomedis menjadi ruang berbagi gagasan, mempertemukan peneliti, akademisi, dan praktisi dari berbagai latar belakang. Pusat Riset Biomedis turut berperan dalam kegiatan penanggulangan bencana melalui dukungan keilmuan dan keterlibatan langsung sebagai relawan. Inari Expo menjadi ajang berbagai karya dan teknologi diperkenalkan kepada publik sebagai bentuk kontribusi riset dalam mendukung pengembangan ilmu pengetahuan. Pusat Riset Biomedis mendukung pelaksanaan olimpiade dan kompetisi ilmiah sebagai wadah pengembangan prestasi, inovasi, dan daya saing generasi muda di bidang riset dan sains. Kerjasama dan kolaborasi menjadi bagian strategis dalam pengembangan Pusat Riset Biomedis. Melalui sinergi dengan berbagai mitra, baik dari kalangan akademik, pemerintah, maupun industri, Pusat Riset mendorong penguatan riset biomedis yang inovatif dan berdampak bagi peningkatan kesehatan masyarakat. Penerimaan calon pegawai negeri sipil di Pusat Riset Biomedis. Momen kelulusan sivitas akademika menjadi penanda capaian penting. Setiap lulusan membawa harapan, pengetahuan, dan kontribusi baru untuk masa depan. Rapat kinerja Pusat Riset Biomedis menjadi wadah evaluasi atas capaian dan pelaksanaan program yang telah berjalan. Melalui forum ini, strategi dan langkah perbaikan dirumuskan untuk meningkatkan kualitas kinerja dan pengembangan riset ke depan. Perjalanan ini menjadi fondasi untuk melangkah ke tahap berikutnya, dengan semangat kolaborasi, integritas, dan inovasi.
Tes. Kedengeran nggak? Tes. Ada.
Iya, kedengeran, Mas. Suara saya terdengar tidak ya, Bapak Ibu?
Jelas, Mbak.
Oke, makasih.
Mbak hari ini sesuai schedule aja ya. 1.50 berarti ya.
Oke, 1 menit lagi berarti ya?
Saya sudah line PI-nya bentar lagi, 5 menit lagi kayaknya, bentar ya.
Oh, Len, oke.
Nanti kalau misalnya PI-nya sudah muncul di screen, dimulai saja nggak apa-apa.
Apanya, Mas? Maksudnya gimana?
Kalau PI-nya sudah muncul di screen, pembicaranya, itu kan masih ketutup ya,
Oh,
belum
bentar.
ada videonya. Ya.
Oh, oke.
Mbak Rini, bentar ya Mbak, ini PI-nya sepertinya ada ini,
Oh ya, oke.
soalnya dia live.
Ada
Iya,
trouble ya?
tunggu sebentar ya Mbak ya.
Oke, baik.
Iya, ada trouble sepertinya. Tunggu ya.
Ya.
Oh, udah. Ini. Halo, Profesor. Halo, halo, Prof.
Muti.
Ah, long time no see. How are you?
Good.
Nice to meet you again.
Yeah, nice to meet you. So everything's
I'm
okay.
sorry. I'm sorry bothering your time. Yeah.
No problem, no
Yeah.
problem.
To give your talk. Yeah.
No problem, no problem. Yeah, my pleasure.
Okay. 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 Maybe we can start, Miss Arini. Yeah. Because the speaker already here.
Okay. Okay, we will begin. Good afternoon, distinguished participants. Welcome to the webinar entitled Advancing Precision Oncology Through Multiomics Approach. Before we begin, kindly pay attention to the following webinar guidelines. Please keep your microphone muted throughout the webinar. Second, participants are encouraged to use the official webinar virtual background. Please fill in the attendance form shared by the committee. Participants who wish to ask questions during the discussion session may use the raise hand feature or submit their question through the Zoom chat box. Thank you for your cooperation and attention. The webinar will begin shortly. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Distinguished Head of the Research Organization for Health BRIN, Professor Niluh Putu Indi Darmayanti. Distinguished Head of the Center for Biomedical Research, BRIN, Professor Sunarno. Our distinguished speaker, Dr. Xin Yu Chen, Associate Research Fellow, Institute of Biomedical Sciences, Academia Siddika, Taiwan. Our respected moderator, Bugiratno Budiarto, PhD, distinguished researchers, academics, students, colleges, and participants joining us online. Welcome to today's webinar entitled Advancing Precision Oncology Through Multiomics Approach. On behalf of the Center for Biomedical Research Organization for Health BRIN, we warmly welcome all participants joining us via Zoom meeting and YouTube live streaming. We hope today's webinar will provide valuable scientific insight and foster future collaboration in biomedical research, cancer biology, and precision medicine. Ladies and gentlemen, to begin the event, let us sing our national anthem, Indonesia Raya. Ladies and gentlemen, to officially open today's webinar, may we kindly invite Professor Niluputu Indi Darmayanti, Head of the Research Organization for Health BRIN, to deliver her welcoming remarks. Professor Indi, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon to Honorable Professor Dr. Sunarno, as the head of the Research Center for Biomedic, distinguished speaker, Dr. Sin Yu Chen, and college researchers, lecturers, students, and all participants. On behalf of the Research Organization for Health BRIN, I am pleased to welcome you to this webinar, Advancing Precision Oncology through Multiomic Approach. We are honored to have the Dr. Sin Yu Chen with us today, and I would like to express our sincere appreciation for his willingness to share his expertise and experience. This webinar reflects a BRIN commitment to strengthening biomedical research, broadening scientific insight, and fostering collaboration with the international partners. Today's topic is highly relevant as the multi-omics approaches are becoming increasingly important in understanding tumor heterogeneity and the tumor microenvironment, as well as the prognosis and response to therapy. The integration of the genomics, transcriptomics, proteomics, and single-cell analysis opens important opportunities for biomarker discovery, patient stratification, and the development of more precise cancer treatment strategies. We believe that this webinar will not only enrich our scientific understanding, but also encourage a meaningful discussion and future collaboration in precision oncology research. I would also like to extend my appreciation to the organizing committee and all the participants for making this event possible. I wish you all a fruitful and inspiring session. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Indi, for the inspiring remark and for officially opening today's webinar. Ladies and gentlemen, before we proceed to the scientific session, we will have a virtual group photo session. We kindly invite our distinguished guest, speaker, and moderator to turn on their cameras. Please look at the camera and smile. Ready? 1, 2, 3. Okay, thank you very much. Ladies and gentlemen, the next agenda is the presentation and discussion session, which will be moderated by Bugi Retno Budiarto, PhD, researcher at the Center for Biomedical Research Organization for Health BRIN. To Dr. Bugi, time is yours.
Okay. Thank you, Mr. Arini, and also thank you to Professor Indri for her insightful opening remarks. So good afternoon, everyone. So before we proceed to the main agenda, it is my pleasure to introduce our speaker today. Dr. Chen is an associate research fellow at the Institute of Biomedical Science, Academy Sinica, Taiwan. He is a very famous researcher in the field of single-cell approach, and he has received many awards, both from the Taiwan government and also international, for his contribution to the field. And also, Dr. Chen has made significant contribution to the biomedical research, and his work has been widely published in leading peer journals such as Science, Nature, Cells, and so on. Until now, he has 41 outstanding scientific publication, and you can see in his IBMS portal. We are honored to have him with us today and look forward to learning from his expertise and also insight. Please join me in welcoming Dr. Chen. So Dr. Chen will give talk around 1 hour. Following the presentation, we will open the floor for a Q&A session for 15 minutes. Dr. Chen will deliver his talk with title as seen soon in the screen. So without further ado, Dr. Chen's screen is yours.
So, do I share my screen? Can
All
you see
good.
my screen?
All good. Yes. All
Okay,
good.
okay.
Go ahead.
And also,
Go ahead.
you can see my cursor, right?
Yes, definitely.
Okay. So first of all, I would like to thank the organizing committee to give me the opportunity to share what we have learned and what's current research in the lab. And of course, as you probably know, like Buji is my first TIGP student when I start my lab 8 years ago. So in addition to try to introduce what we are doing, I also want to thank Buji for her hard work, for his hard work in the lab. And of course, since he's my first TIGP student, I will try to make sure he has a very successful career. So I will do whatever I can do to help his career as well. Okay, so so let's start the topic of today. So the title is Advancing Precision Oncology Through Multiomic Approach. But I have a subtitle, because right now probably you have read a lot of paper, especially the high-profile journal paper. They all use single-cell platform or single-cell analysis. So there are a lot of fancy images or fancy figures. But today I would also want to emphasize that the single-cell analysis is not just profiling, because if you just treat it, consider it as a way to generate a lot of fancy figures, then that definitely not the good things about a single-cell analysis. So I will talk about 3 stories in the lab, which we try to not just using single-cell analysis, but try to push more to study about the mechanism and also even do some manipulation. So hopefully in the end of the talk, I can convince you that single cell analysis is just the beginning, but there are much more hard work you need to do after the analysis. So, of course, you probably hear this kind of story a lot, like the development of the microscope by Hooke actually lead to the observation of the bacteria, and which eventually lead to the discovery of the penicillin, which are used to treat the bacterial infection. So as a training platform people at Stanford, actually, we are interested in developing new tool because we believe with the advancement of the new tool, actually, you can make some observation that cannot be observed before. And from that on, you can start to form your own hypothesis and also design your experiment, eventually design the treatment to treat the patient. So when I was at Stanford, actually, we are working on a lot of single cell platform. So, but those platform are just the tool development. It's the first of everything. Start from the tool development, you would like to use those new tools to make some interesting observation and form hypothesis. And from the hypothesis, you design more experiment to figure out what's the mechanism, and especially what are the causal relationship between the outcome and the cause. And eventually, if we can identify the key causal relationship, we can try to manipulate the cause of the disease, so we can make a new treatment based on the causal relationship we identify. So when I was at Stanford, actually, we are working on a lot of single cell platform, try to develop new single cell platforms. For example, Codex and CyTOF are two single cell platforms which have been developed in Dr. Gary Nolan's lab. So as you probably also heard about these two platforms, so the CyTOF is using antibody-based approach to reach single cell proteomic. So it's just like a flow cytometry, which use fluorescence to conjugate antibody. But in CyTOF, we are using metal, heavy metal to conjugate antibody and use mass spec to detect the protein expressed on individual cell. And Codex is another platform. In addition to to measure the protein expression in solution, we are able to study the multiplex protein information in a single tissue section. So basically, it's a spatial proteomic approach. Of course, right now you heard a lot about single cell RNA-seq, like from 10x or different platform. There are a lot of high throughput single cell transcriptomic platform. So of course, we also did a lot of this kind of analysis in the lab, and also ATAC-seq. More importantly, we also set up the system to probe the 3D genome structure, such as HiChip, which can be used to identify the enhancer-promoter interaction that happen in 3D. Those are the already existing platform we try to set up in the lab. In addition to that, we are also interested to develop new tool which haven't been developed before. So of course, all these are, as I mentioned, all these are just the beginning, the tool development. So of course, we will try to apply all these tools, both existing platform and also new tool, to connect it to a real world situation. For example, try to understand more basic biological principle, and also in the context of clinical setting, in order to solve clinical unmet need. So let me start with the first story I want to tell you. It's about T-cell exhaustion. So of course, I think probably a lot of you have heard of immunotherapy, especially the immune checkpoint inhibitor, which have revolutionized the cancer immunotherapy field. So in the underlying biology process, basically people have identified like the T cell after sustained antigen stimulation, you will undergoing just like a spatialized differentiated trajectory called T cell exhaustion. So from naive T cell, you will go through progenitor exhausted T cell and eventually become the terminal exhausted T cell. And through this differentiation trajectory, people have identified some very important receptors that are upregulated during this process, including PD-1 and TIM3. So by blocking this PD-1, PD-L1 interaction, actually the people can reactivate the T cells function, so they can be broadly applied to different cancer type. And with both basic research and clinical observation, right now we have known that the response to immune checkpoint inhibitor, such as anti-PD-1 and PD-L1, actually the progenitor T cell is the population that is responsible for the response to therapy. And also we know a lot about the process, which is just like a typical differentiation program. There is a chromatin remodeling process happen from the progenitor T cell all the way to terminal exhausted T cell. So back then, when I just done my lab, actually we are interested in this process, especially there are so many checkpoint inhibitors. So we just ask whether there is any checkpoint for all these checkpoint receptors, just like the traditional developmental biology. You always have like a Waddington landscape, and you have some cell fate determination step that will determine what are the cell fate the cell will proceed. So in order to address this question, actually we start with an observation where we are using single cell analysis using the single cell RNA-seq data, which we collaborate with Dr. Huang Yanzhong, also from Academia Sinica. So we took the single cell RNA-seq data from both human and mouse and rank the exhaustionness of the T cell. And interestingly, we can find that, as you see here, the x-axis is the ranking and y is the exhaustion status. So you can clearly see there's a plateau or a transition of the exhaustionness from the non-exhausted to the terminal exhausted. So we are hypothesizing that this is just like the typical cell fate determination process, where there's a critical period and as a cell fate determination step, and after you reach certain threshold, the cell decide which cell fate you will take. So eventually the cell fate will be determined. In this case, is a terminal exhausted. So we hypothesize this critical period, there are some biological process that's important to determine the cell fate. So we do simple differential equation to try to find the point where the slope change dramatically, and use that to define the critical period and try to identify the biological process involved. By doing this, interesting, you can find that in addition to some of the well-known pathway, we also identify unexpected pathway. For example, DNA damage response, chromosome organization, and the proteasome pathway that are also upregulated during this critical period. It's not just in the mouse, but also in the human. So, but that's just one of the observation, and also in one of the cell line or a cell cancer type. So since these two, like a DNA damage response and chromosome organization, are highly enriched, and so that's why we decide to proceed and move on to study whether they are the really driver or the checkpoint of the checkpoint receptor. So as you can see here, we are starting from observation and then from a hypothesis, the DNA damage and chromosome organization are the checkpoint of a checkpoint receptor. Of course, it's just a beginning. So back then, Buji just joined my lab, so he's very interested in this kind of approach and want to know more. So with his help, actually, he tried to collect all different kind of both mouse sample and also different cancer type, and try to reanalyze all the data to make sure our observation is not just in one cancer type, but as a general pan-cancer phenomenon. So as you can see here, if we group all the T cell into non-exhausted, progenitor exhausted, and exhausted, you can clearly see across different cancer types. Actually, you can see the progression of the DNA damage response and also the chromosome organization response. And then, of course, this is just RNA, so you might ask about how about protein, especially DNA damage response is it's well known. There are a lot of protein involved. So another PhD student, Gordon Lee, who have using the CyTOF, the single-cell proteomic platform, and indeed he also noticed that at different time point, like the day 0, from day 0 to day 8 in vitro, actually he can observe the phospho-ATM, which is a key DNA damage pathway that are upregulated during the process. Similarly, the chromosome organization pathway like a cohesin, SMT3, which are also upregulated after T cells are activated, indicating that the observation we made in the RNA is also there, like also hold true at the protein level. But of course, that's a in vitro observation. So we then collaborate with Dr. He Bingzi in Switzerland, and he also applied the in vivo animal model, trying to figure out whether the T cell after sustained simulation, the DNA damage response is upregulated. So he performed a fluorescence barcoding tracing experiment, and you can see here, like with the time, actually when the T cells are infiltrated into the tumor, there's accumulating of the gamma H2AX staining, which indicating the DNA damage response, and also the increased level of the tox, which are a well-studied T cell exhaustion master regulator. More interestingly, he also found that after the the immune cell infiltrated into tumor, actually there is an interesting mitochondrial dysfunction as shown here, where the mitochondria potential is decreased, indicating that after T cell infiltrating the tumor, there are some mitochondrial dysfunction in the tumor environment. When he sorted out the mitochondrial dysfunction subset, actually he can identify that the nucleotide metabolite are decreased in that population as a hint of the potential driver of the DNA damage. We also work on more mechanistic study, try to figure out in the environment or in after T cells are undergoing sustained simulation, what are the origin or source of the DNA damage. And as you can see here, like after T cell are sustained simulation, the replication fork of the T cell actually is shortened, just like when you treat it with the HU, which deplete the nucleotide pool. suggesting that after T cells undergo sustained stimulation, there is a replicative stress undergoing that will interfere with the progression of the replication fork. So right now we come up with a hypothesis where the persistent antigen stimulation will trigger the mitochondrial dysfunction that lead to the depletion of the nucleotide pool. At the same time, the persistent antigen exposure actually trigger the proliferation of the T cell, which induce replication stress. And altogether, these two will lead to the DNA damage response we observed. But you might ask, since those as I mentioned now, like we observe the DNA damage response, and we identify mitochondrial dysfunction and replicative stress as a potential origin of the DNA damage. But you might ask, whether that's a cause of the T cell exhaustion, or it's just a correlation or association with the T cell exhaustion process. So to address this, we need to do some loss of function study. For example, in this case, we are trying to use the ATN inhibitor, which we observe is upregulated in our profiling. And when we block the T cell with the ATN inhibitor, and we can clearly see the increased population of the TCF7 positive and TOX positive population, which are the surrogate marker of progenitor T cell, indicating that the ATM inhibition actually can stop the progression of progenitor exhausted T cell. We can also see that after the ATM inhibitor treatment, actually the PD-1 and TIM3 expression is suppressed, indicating not just the progenitor T cell is increased, but also the terminal exhausted T cell is reduced. Furthermore, we are interested in what happened after the DNA damage. And probably you also heard about the decompartment, which had been published 3 years ago, showing that the DNA damage can trigger the 3D reorganization. So we are trying to do the same approach in the T cell. And indeed, we also see the same. Indeed, we also see the same setting where the repeat antigen stimulation, the chromosome enhancer promoter interaction is upregulated in the TIM3 region. But when we treat it with the ATN inhibitor, actually the increased enhancer promoter interaction is suppressed, indicating that the 3D genome is really reorganized during the T cell exhaustion process, and DNA damage pathway is responsible for the reorganization of in the T cell exhaustion. Furthermore, when we get the knockout of the chromosome organization factor, actually you can clearly see in the knockout animal, the double positive TIM3 and PD-1 population is suppressed in the chromosome organization knockout, indicating that the chromosome reorganization is not just the association, but it's a causal relationship for the T cell exhaustion progression. So to summarize this part of the research, actually we identify the DNA damage response, so DNA damage pathway, as a checkpoint of the checkpoint receptor progression. But then, since, as I mentioned in the beginning, we want to start with observation to find a causal relationship, and after that, we would like to see whether there is any new manipulation that are inspired by our study. So the next question is, how how do we manipulate the DNA damage pathway in T cell, especially as you probably know a lot about the DNA damage response or DNA pathway that are focusing on the tumor. So it will be difficult to find a specific manipulation in T cell using the current existing drug. So to solve this problem, actually, if you remember, when we do the profiling, actually, in addition to DNA damage response and chromosome organization, actually, we also find out the redox process is one of the pathways that are highly upregulated together with the DNA damage response. So we then think about whether we can profiling the redox process in T cell exhaustion process and try to identify novel or key regulator of the redox. And we intend to manipulate the DNA damage pathway through the redox process. But back then, when we try to profile the T cell exhaustion at the single cell resolution, actually we find out there is no good tool. The current existing tool with fluorescence probe is just measure the global level of the redox level. And that indeed gives us single cell resolution, but it's just a global level of the redox. It didn't provide us any molecular information. And if we want to know about the molecular information, we can perform a mass spec analysis. But the problem for mass spec analysis is there is no single cell resolution. Means you need 100 millions of cells in order to identify the molecule regulated by the redox. So we decide to, since there is no tool available, then we are since we have the specialty expertise to develop new tool, we decide to take the challenge and try to develop the single cell redox profiling platform. So another PhD student, each in the lab, is taking this job and screen more than hundreds of antibodies related to the redox pathway according to the mass spec information. And it turned out, it came up with a more precise panel with more than 40 markers at a single cell resolution together with the immunophenotyping marker. With this tool, we try to look at the T cell exhaustion and try to see what are the pathway upregulated and downregulated. Especially, we try to focus on the pathway that link to DNA damage response. So as you can see here, the x-axis is the time, the pseudo time, and the y, all these are the different markers related to redox pathway, and the red indicating that the level of expression is upregulated. So you can clearly see that after the T cell are stimulated, actually the redox pathway is highly dynamic. Some pathways are upregulated in the beginning and then going down, and followed by other pathways. So when we do the Galton analysis, actually, indeed, we find DNA damage response upregulated in the terminal exhausted state, as similar to what we have observed before. But more interestingly, we also noticed that right before the DNA damage pathway, actually there's another protein folding or translation related redox pathway are upregulated. And interestingly, following this period, this upregulation of protein folding redox pathway, when you go down, it comes out with the traditional cellular response to ROS. So we are interested for this ER or translation-related pathway. And we try to manipulate this pathway to see whether actually that's associated or correlated with the DNA damage response, and also whether we can use manipulate this process to manipulate the T cell exhaustion progression. So that's interest. We use the one of the inhibitor of the pathway called APX2009, which block the APE1 enzyme. As you can see here, in the acute state, if there is no treatment, all the T cells are functionally represented by the double positive of interferon and TNF. And when we block the T cell with the APE1 inhibitor, actually the function of the T cell is suppressed. And to demonstrate this suppression is redox related, so we add the anti-oxidation, N-acetylcysteine. And interestingly, we can demonstrate that the phenotype can be rescued, indicating the APE1 inhibitor regulate the T cell in the redox-dependent manner. So all these are about in vitro. So we then try to see whether all this observation, the dynamic regulation of the redox in T cell, also hold true in the patient. So we collaborate with Dr. Tang Jilu from NTUH, where he helped us to collect a time course or sample of the CAR T cell. And for a clinician, he is interested in the persistency of CAR T, because in the clinical setting, he observed patients after the CAR T therapy. Some of the patients actually have higher CAR T after 3 months, but some of patients after 3 months, all the CAR T are almost gone. So he is interested what are the factors, or whether we can predict which patient will have more CAR T in vivo in the long run. So when we apply the single-cell redox platform to this cohort of the patient, and as the figure show, you can see like the x-axis is the day after infusion, and y-axis is the percentage of CAR T that are with active redox status. So you can clearly see like all the patients in the beginning, because the CAR T, the manufacture of CAR T require activation. So you can clearly see all the patients are highly active in the redox pathway. But with time, some of the patients, the redox activity gradually going down, but some of the patients still have the sustained redox activity. And interestingly, when we correlate this information with the clinical data, we find a very clear negative correlation result, where with the patient that have decreasing redox activity, actually their CAR T are more persistent in the long run, in compared to the patient if their CAR T still with high redox activity. Actually, their persistency is poor. So based on this, I hope I can convince you, like in the first part of the story, we demonstrate that DDR is a checkpoint of the checkpoint receptor. And second, redox regulation represents a potential target to restrain the DDR during the T cell exhaustion progression. So of course, people are excited about T cell due to the immunotherapy, and that's why people are with the successful story of the T cell exhaustion, people are thinking about whether there are other exhaustion as well in other immune cells. So the second story I'm going to tell you about is related to the NK exhaustion. So you probably remember this figure, the left figure before, like the T cell after sustained simulation, they will have a differentiated trajectory through progenitor exhausted and terminal exhausted. But for NK cell, the story is completely different because for each T cell, they have a unique T cell receptor. So you can easily trace the progression or the difference after T cells are activated through the T cell receptor. But NK cells are more heterogeneous, so you can consider it as if there's a multiple origin for NK cell. And furthermore, there is no NK receptor for all the NK. Actually, all the NK have a combination of different activating receptor and inhibitor receptor. So that makes the tracing very difficult. That means if you stimulate NK cell, maybe you only stimulate a certain percent of NK cell, and they are in the different origin of the differentiation landscape. So you can imagine once you stimulate NK cell, some of the subset of NK cell are going down, but some of them stay in the origin. So that makes the profiling very challenging and difficult. And of course, since it's very difficult to trace in the progression of NK stimulation, so we also don't know whether there's a similar chromatin remodeling happen during the NK exhaustion process. So that's why not just our lab, when we try to find a PD-1-like receptor in NK cell for NK exhaustion, we cannot find any NK receptor that just like a PD-1. But interestingly, through some of the clinical study, for example, when we collaborate with Dr. Lin Guoyi and Lin Yilin to profile the COVID-19 patient, and also when we collaborate with Dr. Zhong Guibin in NTU Hospital to profile the nosocomial infection cohort, actually we find out the cytolytic activity and especially the metabolic status of the NK cell is correlated with the clinical outcome. That suggests maybe the NK exhaustion, maybe it's challenging to find a single receptor for NK exhaustion, just like PD-1. But maybe the intracellular metabolic status is the checkpoint or the exhaustion checkpoint for NK cell. So with that observation in mind and hypothesis in mind, we start to design experiment, try to figure out whether the metabolic status of the NK cell is a checkpoint of NK exhaustion. So to address this question, we collaborate with Jack Hu in Academia Sinica, where he have a system to gelated the cell, the tumor cell, such that the gelated cell, they don't have any metabolic activity, but they have the intact cell surface. So immune cell can still interact with the gelated cell, but actually the cell is dead. So by using this platform, we try to see whether the metabolic activity of the tumor cell within the tumor microenvironment will change the exhaustion status of the NK cell. So we call it's a gelated cell system. So as you can see here, the y-axis is killing efficiency, and the red line represents the NK growing in the gelated tumor system. You can clearly see that across different donors, all the NK expanded with the gelated system. Actually, they have much better killing efficiency. And this better killing efficiency, actually, you can see it's conserved, or you can across a different cancer cell type, indicating that if NK is expanded or activated in the environment without metabolic activity from the tumor cell, actually the NK will not exhaust. So we also confirm this finding with the in vivo animal model showing that actually the GC, gelatin cell expanded NK cell, have much better tumor control activity, tumor control ability. But then we want to ask, what are the key factors in the environment that can drive the metabolic related NK exhaustion? So we perform a mass spec of the soup of the environment and identify that the lactate in the media, in the environment, is highly upregulated in the live tumor cell versus the gelated tumor cell. So we do an in vitro experiment to demonstrate that indeed, when we add the lactate in the environment, we can see a suppression of the killing function of NK cell. And furthermore, even when we knock down the transporter, making the NK cell not able to import the lactate from the environment, we can see the suppression or the exhaustion phenotype is suppressed by the knockout. Then we want to ask, what happened after the lactate is imported into the NK cell, and also whether there is any epigenetic reprogramming after the lactate is imported into NK cell. So when we perform the cytol experiment, actually we can clearly see that when NK cell is cultured in the high level of lactate, can clearly see a shift of the epigenetic level such that the acetylation is downregulated, but at the same time, another histone modification, histone lactylation, is upregulated in the presence of the lactate. So with that, we show that lactate is the factor that can drive the histone acetylation, hypoacetylation, and hyperlactylation. But how does this epigenetic switch trigger the NK cytolytic suppression? We then go ahead to do RNA-seq and also high-chip to show that actually the NK cell mediated cytotoxicity is the pathway that are upregulated, are downregulated by the lactate treatment. And when we perform the high-chip experiment, actually we can clearly see that some of the important effector functions like perforin, the enhancer promoter loop, is significantly suppressed by the lactate treatment, indicated by the red arc. Interestingly, when we overlay the histone lactylation time take, the peak, actually we notice that the peak of the histone lactylation colocalize with the loop of the acetylation, indicating that the histone lactylation induced by lactate actually modulate the chromosome interaction, enhancer promoter interaction in NK cell, especially in the cytolytic genes. But all these are in vitro observation. Then we ask whether all this epigenetic switch happen in the real patient. So we collaborate with Dr. Donghong Su from NTUH. He helped us to collect the sample from HCC, hepatocellular carcinoma patient, where we have the normal, adjacent normal part, the junction part, and also the center part, which are more hypoxia. So when we perform the the mass cytometry, actually we can also clearly see from normal to junction to cancer, there is a trend from up to low. And from junction to cancer, actually there's a trend from left to right. And we're interested in what actually are the subsets that are enriched in the cancer and also junction. Interestingly, we find out that the trend match very well with the epigenetic switch we observed in the in vitro experiment, where the acetylation is downregulated when we profile it from the junction to cancer, and at the same time, the histone dactylation is upregulated when it is in the hypoxia region or the cancer core region. Demonstrate that it's not just in vitro, actually in vivo, we also observe the epigenetic switch, the hypoacetylation and hyperlactylation in the NK cell in tumor. But still, we still don't address the mechanism why lactate import can trigger the epigenetic switch. So we think about the fundamental biochemical reaction, what we have learned before. The lactate, you probably remember in the textbook, the lactate in the cell is generated through the pyruvate through the LDH as an enzyme. And this step actually is the key enzyme for replenish NAD. So we think, we reason that if there are more lactate in the NK cell imported from the environment, supposedly that will trigger the reaction to the opposite side. And that in the decrease of the pyruvate to lactate will make the replenish of the NAD decrease. So indeed, that's what we observed with the mass spec experiment, where we indeed see the decreased level of NAD plus with the lactate treatment NK. More importantly, we reason that all the NAD dependent, all the biochemical reaction require NAD as a cofactor will be inhibited. So you can clearly see that these three steps in the TCA cycle require NAD as a cofactor, and all of them, the substrate is accumulated, and the end product is downregulated, indicating that the metabolic process in the NK cell, all the NAD-dependent biochemical reaction is inhibited. It's not just in the TCA cycle. In the glycolysis, it's the same. Like the reaction requires NAD as a cofactor. The substrate is accumulated, and the product is decreasing. So we then want to demonstrate the causal relationship by adding more NAD as a rescue. And you can clearly see that with the treatment of the lactate, the suppression of the cytolytic function is rescued by NAD, also by the NAD precursor, NR. So indicating it's not just correlation, but it's a causal relationship. So because of the limit of time, so I won't go through too much mechanistic detail, but with more like an imaging study and also biochemistry study, right now we figure out the decrease of the NAD actually can affect another NAD-dependent process called pyrylation, and that lead to the decreased translocation of an enzyme called PDH. And the pyrylated PDH actually is the enzyme responsible for the synthesis of acetyl-CoA in the nucleus. That's why in our NK study, we observed the hypoacetylation in the NK when we treat it with lactate. But at the same time, the increased level of the lactate will increase the lactyl-CoA, which lead to the more histone lactylation in the nucleus. And that's why we see this hyperlactylation, hypoacetylation epigenetic switch in the lactate-treated NK. So in the second part of the talk, we try to study the NK exhaustion, and we find out that the metabolic epigenetic switch triggered by lactate could be a potential mechanism of the NK exhaustion. And finally, I would like to introduce another story why we focus a lot on manipulation, not just observation, try to identify causal, but try to design a new drug. So you probably all know about the anti-cancer immunity, where the innate immune, they will recognize the tumor cell and release the tumor antigen as well as the damage-associated molecular pattern, which can recruit antigen-presenting cell, including the dendritic cell. And the APC antigen-presenting cell will uptake the tumor antigen and migrate to lymph node to present antigen to T cell, such that the adaptive immune from T cell will be activated, and the activated T cell can migrate to the tumor and to kill the tumor. So those are the typical anti-tumor immunity cycle. So inspired by the CAR-T design, where artificially we fuse different signaling domain within one cell. So we are asking whether we can artificially create a circuit like this by creating an artificial interaction between dendritic cell and innate immune cell. So when we try to design or try to get the candidate that can perform this kind of dendritic cell engager function, we also get the help from Buji, where he, with his expertise in bioinformatic analysis, we are able to identify two unique cell fate from the pancreatic cancer sample we profiled. And you can clearly see that from the non-malignant cell, actually there are two different differentiation trajectory. And when we look at the signature of these two branches of the tumor progression, actually you can kind of see that the sialic acid-related pathway are upregulated in one of the branch. So we then, based on those observations, and then project it to the spatial transcriptomic data, and we can identify that those sialic acid upregulated cancer types, actually they are colocalized with the dendritic cell, as shown with the spatial transcriptomic data. So that makes us think the sialic acid and is potentially inhibitory pathway or inhibitory ligand receptor interaction happen in vivo to inhibit the dendritic cell activation. So we then try to see whether other cancer types also have the upregulation of sialic pathway. And indeed, you can see here, not just the pancreatic cancer, but also triple-negative breast cancer, non-small lung cancer sample. You also can clearly see that the sialic pathway are upregulated in the malignant cell compared to the normal epithelium control. As you probably know, that sialic acid on the receptor for sialic acid is called ciclet. And across all these three different cancer types, we can also see that the ciclet 10 is one of the ciclet that are upregulated in the dendritic cell. So that indicating that in all these three cancer types, there is sialic acid expression in the tumor, and at the same time, there is a cyclic expression in the dendritic cell. So that makes us figure out whether we can design a drug that blocks this cyclic and sialic acid interaction in vivo. So that come up with our design. We try to design a bifunctional stimulant which can block the cyclic on the tumor, but on the other end, activate dendritic cell and bring these two together. So with that in mind, actually a student me in the lab trying to make this kind of cyclic GM-CSF fusion cytokine. And you can see that by fusing these two together, actually the function of cyclic is still there. Like the fusion cytokine can still bind to the cancer cell. And with the biofunction assay, you can clearly see that even the fusion cytokine, they can still have the GM-CSF activity. In the in vitro study, we can also see that the incubation or the bifunctional cytokine actually can activate a dendritic cell. In subsequently, the co-culture of the activated dendritic cell actually can activate T cell more. Furthermore, when we do the image study, we can clearly see that the bifunctional cytokine can bring cancer cell and dendritic cell together and also activate them to facilitate the colocalization, the phagocytosis of the cancer cell by the dendritic cell. When we perform the in vivo efficacy study, we can clearly see that the tumor cell express the bifunctional cytokine. Actually, half of the animal become tumor-free in the pancreatic cancer animal model. More importantly, when we re-challenge those cancer-free animal, actually the tumor cannot grow anymore. And this kind of observation also in the 41, which is a breast cancer model, when we re-challenge, we also see tumor-free when we express the SG11. And when we try to re-challenge 41, actually none of the animal can, the tumor can grow, showing that the bifunctional cytokine is not just provide the protection, but also have the anti-tumor memory. We then make it more therapeutic, mimic the therapeutic condition by injecting the recombinant protein, and you can clearly see that by itself or with combination of checkpoint inhibitor, actually you can also get certain % of the tumor-free animal. And when we do more mechanistic study to deplete the dendritic cell and T cell, we can also clearly see that the effect of the bifunctional cytokine require dendritic cell and T cell as we expected. So of course that demonstrates the efficacy of the bifunctional cytokine. As I mentioned in the beginning, we try to build an artificial anti-tumor circuit by linking the innate immune and dendritic cell. So right now we are engineer the gamma delta T and also NK cell with the bifunctional cytokine, hoping to make the innate cell that kill the tumor, release the antigen, at the same time activate the dendritic cell in the same location. So we are collaborating with NTU Hospital, and right now we are able to make a clinical grade gamma delta T cell that can traffic to the tumor region very well. Of course, we try to not just work in this in the lab, but we try to move it in the clinic. So we form a team to not just doing research, but also doing some business development and also try to proceed into the clinical trial. So with that, I think I have go through all three stories in the lab, but what we are doing now, what you might interested in what we are going to do in the future. So of course, as I mentioned, we are working on T cell exhaustion. We are working on NK cell exhaustion. There are so many other immune cells, but we are interested in whether there is any other mechanism of immune exhaustion in addition to NK and T. And also, when people are using single cell platform, usually people will challenge the cohort size because usually all the analysis is only performed profiling in dozens of patients. But we're in the real world, there are so many different people. There's so much heterogeneity. So people always challenge whether all those observations can still hold true in the population level. So to address all these questions or concerns, we right now are working toward the population level single cell analysis. One of the large cohort we are working on is collaborating with Dr. Wu Winxun in Kaohsiung Medical University, where we have more than 500 patients, longitudinal patients of the end-stage renal disease. And we perform not just single-cell RNA-seq, but also metabolomic, redox, and epigenetic profiling together with the genotyping. We try to identify the key factors that are, especially in the uremic toxin, that are important to cause the immunodysfunction in the end-stage renal disease patient. From a pilot study, we can clearly see that the redox patterns are different in the hemodialysis patient. And from a follow-up of this patient, actually, we can identify a more specific redox signature in the monocyte, which can be used to predict the sepsis event during the follow-up. So right now, we are analyzing all the data from single-cell RNA-seq and also mass spec, try to identify what are the uremic toxins that can trigger the redox imbalance in the monocyte, which lead to the immunocompromised status in those patients. As a way to demonstrate the single-cell analysis can be even used in the population level. Of course, that's still, you might say, 500 patients is still a small cohort. So that's why actually we are also working on a TPMI cohort where there's 500,000 of the Taiwanese people is collected with and genotyped. So right now there are we have all the genotyping data, genotype data in those patients, and we try to figure out why all those SNP. What are the function of all these different SNP? Because as you can clearly see, some of the previous study we have shown that the regulation of one gene maybe is distant or far away from the location of the gene. So we hypothesize that the current GWAS analysis cannot be used to identify those kind of 3D regulation. So in that part, we collaborate with Dr. Zheng Jiamin and also Ke Taimin and also Gu Lunwei, where we are trying to build a language model to predict what are the SNP that have the distal regulation and try to use this to improve the prediction of the genotyping to phenotype. Of course, we work on a lot of multiplex imaging, but all those are in the research grade. So we want to make it in the clinical setting. So we decide to design a machine just like a coffee machine where you have a capsule and then a machine to automatically make a coffee. So we try to make an antibody cocktail and with a machine that can automatically perform the multiplex imaging in the clinic side, such that the advantage of a multiplex imaging can improve the prediction of the treatment outcome in the hospital. So that's our prototype. And with that, we hope in the future we will be able to translate this research grade finding or instrument into a clinical setting. So finally, that's my take-home message, where the immune dysfunction in the complex tumor microenvironment is very heterogeneous, and different cell types probably have different mechanisms of immune dysfunction. And also, I hope I can convince you that the newly development, the advance in the new technology actually can help researchers to identify or observe something that cannot be observed and form new hypotheses. But of course, the hypothesis is just the beginning. There are a lot of more to do to validate those hypotheses and find the causal relationship and even design a drug. And finally, I would like to emphasize the importance of collaboration, which, like in the acknowledgement, there are so many different collaborators that help us to move on all different projects. There are a lot of collaborators from the clinical side, a lot of collaborators from the bioinformatics side, and also a lot of collaborators with the help of the animal model. With that, I cannot emphasize more about the help from the people in the lab, especially Buji's help, which helped me to basically set up almost the lab like from the scratch. So of course I get funding from the government and also the academic syndicate, and also I have some disclosure about some company related to single cell platform. And with that, I will close my presentation, and any question or any discussion about collaboration is highly welcome. Yeah, thank you.
Okay, thank you, Prof. Chen, for your excellent talk and also the data present and many updates regarding the research that you already ongoing. I'm happy to hear that. So I think we start to the Q&A session. So I think we have like 2 or 5. I think we have like several questions from the floor. The first question is that this is regarding the NK cell study, Professor. So the question is, as we know, it is called the Warburg effect in the cancer cells that increase lactate production, even though not in hypoxic condition. Based on your finding, it causes NK cell exhaustion. What is the best strategy to overcome this phenomena?
Yeah, that's a great question. I think, yeah, that's exactly the message I try to deliver. So observation or mechanism is just a beginning. So we observe lactate trigger NK exhaustion. We also demonstrate it's a causal. We study a mechanism, but how are we going to reverse, right? How are we going to manipulate? That's a key. So that's also the reason we start the project and also start the screening of lactate transport, because we think it's very challenging to block the lactate in cancer cell because there are several LDH inhibitor trial fail. And we also think cancer is too complicated. If you block something, they will, with evolution, some resistant clone will emerge. So it's very challenging. So we think it might be easier to manipulate the immune part. That's why one approach we are doing is we try to identify the importer and try to block the import of the lactate into NK cell. So for example, when we do the screening, we identify the key lactate importer that are responsible for the NK exhaustion. So right now we are trying to design a drug to specifically block that lactate importer. So even if there are lactate in the environment, as long as the lactate is not uptake by NK, then NK is fine. So that's one approach. And the second approach is that, as you probably remember, we are doing a rescue experiment to supplement the lactate, but supplement the NK with NAD. Because we are doing the mechanism study and identify NAD as a key driver. So we rescue the phenotype by supplement NK with NAD. So another approach we are working on right now is using nanoparticle to deliver NAD or NR, the precursor of NAD, try to specifically rescue NK cell. So we think that's will try those approach will be able to rescue the NK cell even if the tumor cell still have the Weber effect or still generate a lot of lactate, but we are still able to specifically target the NK cell without without the manipulation of the lactate in the tumor part. Yeah. So, but that's still ongoing work. So we cannot provide evidence showing that which approach is better or whether it will work in a real clinical setting because the tumor is like it's a very complicated environment. It takes so long to evolve. So we still don't know whether by manipulating NK cell itself will be enough to reverse the entire disease progression. But we hope that can become one of modality that can combine with other immunotherapy to make the immunotherapy stronger.
Okay, I think I hope that explanation will answer the question. So the other question is that in the translational process, Professor, do you have any barrier on that? Any barrier or bottleneck to translate your
Yes, barrier. Yes, I think the question is very good because it's barriers. It's a S, so there are a lot of
Yes.
barrier. It's not just one barrier. So
Definitely.
yes, definitely. So that's why, especially for the clinical side, it's very well established. So and the clinician need to take the responsibility. So everything need to be very well demonstrated and also will fit to the existing pipeline. The more you fit with the existing pipeline, the better. That's why, for example, the one I mentioned to you, the multiplex imaging, that part, actually, we basically collaborate with a lot of clinical pathologists who help us to implement the machine in the hospital side. And especially we want to make it automation because you cannot expect the hospital or clinical side that can train people and then do the staining manually for you. So we need everything automation and standardized without much variation. So that's why we focus a lot about the automation, try to make things very robust, and try to make it fit with the existing pipeline and collaborate with people need this or people already there. That's one approach we trying to push. And another thing, like that's for the diagnostic part. And for the the drug development part, actually, it's also very important to work with the clinician in the early, because for the clinical trial, all these require the clinical team to support. So that's why in the beginning of the project, we all start with the collaboration already. Who is interested in clinical trial and interested in developing new drug? So that eventually, when we get the approval of the new drug development, then there's at least some clinician willing to take the risk or taking the responsibility to move on the clinical trial for us. Yeah, of course, the major barrier for drug development or clinical trial is the money. So
Yeah.
so that's something that's also the reason why initially I moved back to Taiwan, because when I was at US, actually, I also worked with my previous supervisor in a kind of startup company, and eventually the company is acquired by Roche. So I kind of have the idea how the ecosystem is working in the US, but back then the ecosystem in Taiwan is not very well established. But that's the beginning of the biopark in Taiwan. So I know the government is trying to work on that direction, because before that, like when I was a medical student, people always think like you just do research. You don't think about business. You just publish paper. But I think right now the atmosphere is changing. Like the government in Taiwan, at least in Taiwan, is trying to support researchers to move their research become a real product or connect it to the real world. So back then, I decided to move back to Taiwan and then try to set up similar system in Taiwan, try to gradually build up the ecosystem in Taiwan that can make the process from the bench to the clinical site more smoother. Yeah, so I don't know whether it will succeed or not, but at least I can see more funding to support this kind of research, because this kind of research, you won't get you a Nature Science paper, right? A clinical trial, the PK/PD, that's it's not novel, but it's required, and it takes a lot of money. So, but at least right now there are government support to help with that part, that for the regulatory, so we can try to move forward our research finding into pre-IND and even file for the clinical trial. So that's definitely something we are very interested to working, and we will try to push those products into the real clinics. Yeah, but I think a lot of time it's more about the government support and also the industrial. Like you need to have an ecosystem with a strong angel investor, VC support all the way to make it happen. Yeah. So luckily, I think right now the Taiwan government and also the investor in Taiwan is gradually moving toward that direction. Yeah, so that's why we actually luckily can get some money for that.
Yeah,
Yeah.
that's good. Good way.
Yeah.
So, Professor, there is another question. This is about
Uh-huh.
the technical
Uh-huh.
aspect of
Uh-huh.
the single
Uh-huh.
cell. So
Uh-huh.
the question is, how does integrating the omic data will improve interpretation of the single cell result? Yeah. And
Yeah, yeah.
also,
So yeah,
yeah,
so,
yeah,
yeah. That's
yeah.
why we are we are, as you can see in all the project we are working on, we are not just using one platform. We are not just using like a single cell RNA-seq. Remember in the, for example, the DDR project, we start with observation with a single cell, but we validate with a protein level and then epigenetic, because we believe they are like each layer of information, they can complement with each other, and we will get information. For example, the kind run, we can identify the like the interaction between epigenetic and the chromosome, the enhancer promoter interaction. So it's like the more you observed, that can help you to explain your data better and also try to make a more consistent conclusion across different layers of information. Yeah, so that's why we always trying to do multi-layer, multi-omic approach to try to get the most consistent or most robust observation. Otherwise, when you just look at the single cell RNA-seq, it's very easy to get hundred or thousand of gene of interest. But if you combine it with the epigenetic or proteomic, maybe the overlapping of all these will gradually decrease. And those, and even not just across multiomic, we also try to expand it into multiple cancer type, right? So in that case, we were trying to find the one that are consistent across different cancer type, not just one cancer type or just one cell line, right? So all these approaches trying to help you to filter out the noise, try to find the more robust principle that are conserved across different systems. Yeah, so I definitely would try to get as more modality as possible to get the most consistent or robust conclusion.
Okay. So we move to other question. So in
Okay.
your cohort study, right, Professor, you
Uh,
use how to say Taiwanese, right? Taiwanese
uh-huh.
population. So in
Yes.
the future, will you also compare to other population from the Southeast Asia, like maybe from
Yeah.
how to say, yeah.
Yeah. So that's why because all this require a good banking, biobank, right? So,
Mhm.
for example, most of our study, in the exploratory part, like the language model, when we try to build the language model, AI language model for 3D genome in the GWAS study, we start with UK Biobank, European, the United Kingdom Biobank, because that's the most well-established data, the biobank, and the data is available. So we start with that, but then as people in the TPMI, the Taiwanese biobank is showing, there are SNP that are only enriched in Han population. So that's why people start to build their own biobank, start to collect the genotype from particular population, because there are certain SNP that only enrich in certain population. And also that's why in those study, actually, for example, we observed the HBV, the hepatitis B virus, is anti-correlation with the autoimmune disease in TPMI in Taiwanese population. So that means, of course, like diabetes or like high hypertension, all those maybe you got a lot of common SNP or risk variant across UK Biobank and TPMI, but of course there are some unique features that are only observed in your own cohort. So that's why, of course, that need money, but of course that will be. I feel that's an important infrastructure for the biomedical research in different country or different population. Because with that, if you don't collect those information, then you don't know what are the unique feature in your population. So you basically need those information in order to figure out what to explain why there are some prevalence of certain disease in your population, and also that will become a foundation for a lot of research in the country. That, for example, the reason we are working on hemodialysis or end-stage renal disease is because that's the top one disease in Taiwan that costs the most money in our health insurance. So we want to figure out whether we can improve the care of the hemodialysis patient, because that take 1/3 of the insurance money. So if we can improve that, then that will benefit a lot, not just the society, but also the health burden, right? So that's why I think if you, I personally, I don't know how the biobank system in Indonesia, but I think that will be a very important for infrastructure to build. If and eventually probably will benefit not just the research in participate in that project, but also a lot of other research focusing on some specific disease unique to Indonesia. And if they have fundamental infrastructure, fundamental genotype information, that probably can help that kind of research as well. Yeah. So that's why. So that's why we work on the TPMI, the Taiwan genotyping a lot, because we feel that will definitely benefit the society and also benefit the entire country. Yeah.
Okay, okay, Professor. Because the limit of time, Professor, then we will close this session, and I will hand over the next agenda to Miss Arini. Professor, stay.
Okay.
Don't go away.
Okay, thank you very much, Mas Bugi. Maybe you will present the certificate first. Okay.
Okay.
We will present the certificate to our distinguished speaker, to Dr. Sin Yu Chen. Thank you very much, Dr. Sin Yu Chen, and also our moderator.
So should I
Okay.
virtually shake hand and virtually take the certificate?
Yeah, okay.
Okay. Thank you very much. Okay. Okay, ladies and gentlemen, as we come to the end of today's webinar, we kindly remind all participants to complete the attendance form provided by the committee. On behalf of the Center for Biomedical Research Organization for Health BRIN, we sincerely thank you, our distinguished speaker, moderator, and all participants for joining us today. We hope the knowledge shared in this webinar will contribute to advancing biomedical research and precision oncology in Indonesia and beyond. Thank you for your participation. Stay healthy, stay productive, and continue supporting science and information. Wassalamualaikum warahmatullahi wabarakatuh.
Yeah, if you have
Bye.
any
Thank you very
question,
much.
please
See you
feel
next
free.
in the next event. Yes.
to email me or email Buji. Yeah. So if you need more discussion about collaboration or anything, yeah, just email me or email Buji. Yeah.
Sure.
I'm
Okay.
more than
Okay.
happy to support you and help you.
Okay.
Uh,
Good.
uh, uh,
Bye. So, kita bisa lah ini ya, live ya, live itu deh. Oke. Apa?